Friends, today will we discuss about hemoglobin. Introduction. Hemoglobin is the iron containing coloring pigment or matter of the red blood cells called RBC. It is a chromoprotein forming 95% of dry weight of RBC and 30 to 34% of bad weight. Function of hemoglobin is to carry the respiratory gases, oxygen and carbon dioxide. Molecular weight of hemoglobin is 68,000 Dalton. Normal hemoglobin content. Age at birth 22 to 25 gram per dl. After third month of birth 18 to 20 gram per dl. After 1 year 16 to 17 gram per dl. From puberty onward 14 to 16 gram per dl. Sex. In adult males 13 to 15 gram per dl. In adult female 12.5 to 14.5 gram per dl. Different types of hemoglobin. Normal major types of hemoglobin. First type, second composition and symbol, globulin chain, percentage of total hemoglobin. Remark. HbA1 type. Its composition 2 alpha and 2 beta chain. Globulin in this chain 121 amino acid into 2 plus 146 beta chain amino acid into 2. Total hemoglobin percentage 90%. It is start birth to adult. HbA2 hemoglobin 2 alpha chain 2 delta chain. Alpha chain amino acid 141 into 2 and delta chain amino acid 512 into 2. It is less than 5%. It is increased in thalassemia type. HbF fetal hemoglobin, also called fetal hemoglobin, 2 alpha chain and 2 gamma chain. Alpha chain contain 121 amino acid into 2 plus 150 gamma chain amino acid contain. It is present less than 2% of total hemoglobin. It is present 18 week fetal to birth. HbA1c hemoglobin HbA1c. It is contain 2 alpha 2 beta plus glucose chain 141 amino acid plus 2 into 2 plus 146 amino acid beta chain plus glucose. It is present total hemoglobin less than 5%. It is increased uncontrolled in diabetes mellitus. Functions of hemoglobin. Transport of respiratory gases. Main function of hemoglobin is the transport of respiratory gases. First, oxygen from the lungs to tissues. Second, carbon dioxide from tissue to lungs. First, transport of oxygen. When oxygen binds with hemoglobin, a physical process called oxygenation occurs, resulting in the formation of oxyhemoglobin. When oxygen is released from oxyhemoglobin, it is called reduced hemoglobin or ferrohemoglobin. Second, transport of carbon dioxide. When carbon dioxide binds with hemoglobin, carbohemoglobin is formed. The carbon dioxide can be released from this compound. Structures hemoglobin. It contains 2 alpha chain and 2 beta chains. It combined with iron. 4 chain combined with iron. Polypeptide chain, molecular weight and amino acid. First alpha chain 15,126 molecular weight, 141 amino acid. Beta chain 15,866 molecular weight and 146 amino acid in beta chain. Types of normal hemoglobin. Hemoglobin is of two types. First, adult hemoglobin HbA, second, fetal hemoglobin HbF. Synthesis of hemoglobin. Synthesis of hemoglobin actually start in proathoblastic stage during apoapsis for the formation of RBC, red blood cells. Hemoglobin appears in the intermediate normoblastic stage only. Production of hemoglobin is continued until the stage of reticulocyte. Reticulocyte is the last stage for immature RBC. Heme portion of hemoglobin is synthesized in mitochondria, and the protein part globin is synthesized in ribosome. The reaction is stimulated by erythropoietin and inhibited by heme. Synthesis of heme. Heme is synthesized from succinyl CoA and the glycine. The sequence of events in synthesis of hemoglobin. First step in heme synthesis takes place in the mitochondria. 2 molecules of succinyl CoA combine with 2 molecules of glycine and condense 2 from alpha amino linoleic acid ALA by ALA synthase enzyme. Second, ALA is transported to the cytoplasm. 2 molecules of ALA combine to form porphobilinogen in the presence of ALA dehydratase. Third, porphobilinogen is converted into uroporphobilinogen first by uroporphobilinogen first synthase enzyme. Fourth, uroporphobilinogen first is converted into the uroporphobilinogen third by the porphobilinogen third co-synthase enzyme. Fifth, from uroporphobilinogen third, a ring structure called porphobilinogen third is formed by uroporphobilinogen decarboxylase enzyme. Porphobilinogen third is transported back to the mitochondria, where it is oxidized to from protoporphobilinogen 9 by porphobilinogen oxidase. Seventh, protoporphobilinogen 9 is converted into protoporphyrin 9 by protoporphyrin oxidase. 8. Protoporphyrin 9 combines with iron to form heme in the presence of ferrosilate. Synthesis of globulin. Polypeptide chains of globin are produced in the ribosomes. There are 4 types of polypeptide chains, namely alpha, beta, gamma and delta chains. Each of these chains is different from each other by the amino acid sequencing. Each globin molecule is formed by the combination of 2 pairs of chains, and each chain is made up of 141 to 146 amino acids. Adult hemoglobin contains 2 alpha chains and 2 beta chains. Fetal hemoglobin contains 2 alpha chains and 2 gamma chains. It is the structure of hemoglobin synthesis. two molecules succinyl-CoA plus glycine. The presence of ALA synthase convert alpha amino acid ALA. It is mitochondria to transport in cytoplasm. Two molecule alpha amino acid ALA to convert porphobilinogen with the help of synthase enzyme. Uroporphobilinogen first to convert uroporphobilinogen third with the help of CoA synthase enzyme. Decarboxylation to continue porphobilinogen third into convert porphobilinogen third oxidize in mitochondria, then production protoporphobilinogen 9 oxidase protoporphyrin 9 ferrosylate. It is made up of heme. It is mitochondria to cytoplasm. Heme plus globulin both are combined. Heme plus globin formation of hemoglobin. Substances necessary for the hemoglobin synthesis: iron, folic acid and vitamin B12, proteins. Destruction of hemoglobin. After the lifespan of 120 days, the RBC is destroyed in the reticuloendothelial system, mainly in spleen, and the hemoglobin is released into plasma. The hemoglobin is degraded in the reticuloendothelial cells and spilled to globule and heme. Globin is utilized for the resynthesis of hemoglobin. Heme is degraded into iron and porphyrin. Iron is stored in the body as ferritin and hemosiderin, which are reutilized for the synthesis of new hemoglobin. Porphyrin is converted into the green pigment called biliverdin. In human being, most of the biliverdin is converted into the yellow pigment called bilirubin. Bilirubin and biliverdin are together called the bile pigment. Bilirubin enter into the liver and made up of conjugated bilirubin. Mostly bilirubin enter into kidney, formation of urobilinogen, then excrete into urine. Some of the part bilirubin enter into the intestine and formation of stercobilinogen and excrete into stool. It is the destruction of hemoglobin. Destruction of red blood cells after 120 days. Hemoglobin, heme and globulin. Globulin are reutilized. Heme is converted into bilirubin. Bilirubin and albumin circulation and entered into the liver. Liver bilirubin, conjugated bilirubin, mix up of enzyme then called conjugated bilirubin. Conjugated bilirubin, most part of the bilirubin enter into the kidney. Kidney made up of urobilinogen, then excrete with the help of urine. Second part of bilirubin enter into the intestine and conjugated bilirubin, then made up of stercobilinogen and pass out by the stool. Iron metabolism. Importance of iron. Iron is an essential mineral and an important component of proteins involved in oxygen transport. So human body needs iron for oxygen transport. Iron is important for the formation of hemoglobin and myoglobin. Iron is also necessary for the formation of other substances like cytochrome oxidase, peroxidase and catalase reaction. Normal value and distribution of iron in the body. Total quantity of iron in the body is about 4 gram. Hemoglobin 65 to 68%. In the muscles as myoglobin 4%. As the intracellular oxidative heme compound 1%. In the plasma as transferrin 0.1%. Stored in the reticuloendothelial system 25 to 30% of heme or also called iron. Dietary requirements: adult male 10 milligram per day, menstruating woman 18 mg per day, pregnant and lactating woman 40 mg per day. Source of iron: rich source of iron, organ meats like liver, heart and kidney. Good source: leafy vegetables, pulses, cereals, fish, apples, dry fruits. Poor source: milk, wheat, polished rice. Thank you everyone.